Hi, my name is Marisha binti Mansur and I am from D1 M3P3 and this is my teammate for our biology group.
My name is Nur Anis Amira binti Muhammad Fauzi.
My name is Rarul Sakina binti Muhammad Fisul.
My name is Nur Nazur Anisa binti Muhammad Nasir.
My name is Nur Ada Natasha binti Abdul Rahman.
And today we're gonna explain about DNA recombinant in technology. So that's it.
Sequence of DNA containing gene of interest. For example, gene encode for protein which can dissolve blood clots.
The second tool for recombinant DNA is restriction enzyme. Restriction enzyme is enzyme which cut DNA molecule at a restriction site and produces short restriction fragments. Restriction site is the short base pair sequence of 4 to 8 base pair recognized by a particular restriction enzyme. Restriction site contain palindromic, where the same sequence of bases is found on both DNA strands but in opposite direction. For example, GAA-TTC, GAA-TTC. There are 3 examples of restriction enzyme. The first one is EcoR1. EcoR1 produced by E. coli. It recognizes the base sequence of GAA-TTC and makes staggered cut with the sequence, produce sticky ends. The second example of restriction enzyme is SMAR1. SMAR1 produced by Serrata marquescens recognizes the base sequence of 5' CCC GGG to 3'. The cut is made between adjacent C and G, cut straight across the double helix, produce blunt end, straight cut. Third one is BAM-H1, produced by Bacillus amylocufacans, recognize the base sequence of GGA-TCC. It cut between G and G, makes staggered cut, produce sticky end.
The next tool is DNA cloning vectors, which act as transport. DNA molecule which can insert fragment into a cell and replicate in that cells. The characteristics for DNA cloning vectors are able to accept foreign DNA, able to replicate freely, possesses selectable genetic marker, for example is antibiotic. DNA cloning vectors has 4 examples, which is plasmid, bacteriophagy, cosmid, and yak. Plasmid carry foreign DNA into a host cell and replicate there. The capacity for plasmid is 1 to 10 kilobase. The next example for DNA cloning vectors are bacteriophagy. Bacteriophagy is a virus that infects bacteria. The capacity for bacteriophagy is 8 to 20 kilobase. The next example for DNA cloning vectors are cosmid. Cosmid is a hybrid vector which is combination of plasmid and cos gene. The cloning capacity for cosmid is 35 until 50 kilobase pair. The last example for DNA cloning vectors are yak. Yak is a fragment of DNA from the human genome inserted into a yeast cell. Yak has a cloning capacity from 100 to 1000 kilobase pair.
Host cell, a cell which receives recombinant DNA and allow the replication of recombinant DNA. Its characteristic is it's able to receive recombinant DNA through the transformation process. Second, it's able to maintain the structure of recombinant DNA from one generation to another. And lastly, it's able to amplify the gene product from the recombinant DNA. Bacteria is the most commonly used as host cell. The reason is DNA can be easily isolated from or introduced into a bacterium. Bacterial cultures grow quickly, rapidly replicating, any foreign genes they carry, for example, E. coli. Lastly is modifying enzymes. DNA ligase. It joins the foreign DNA fragments containing gene of interest and bacterial plasmid by catalyzing the formation of phosphodiester bond to form recombinant DNA. Tag polymerase. It used in polymerase chain reaction, or the short form is PCR. It replaced the DNA polymerase because of the temperature conditions of PCR. It also to add nucleotides to the 3' end of primer in order to produce new DNA strain. Tag polymerase also can survive in high temperature.
Cloning, which is all about making identical copies of a piece of DNA. And usually it's a piece of DNA that codes for something we care about. It is a gene that will express itself as a protein that we think is useful in some way. Now, you might have also heard the term cloning in terms of the Clone Wars in Star Wars, or Dolly the sheep, and that is a related idea. If you're cloning an animal or an organism, like a sheep, well, then you are creating an animal that has the exact genetic material as the original animal. But when we talk about cloning and DNA cloning, we're talking about something a little bit simpler, although as we'll see, it's still quite fascinating. It's identical copies of a piece of DNA. So how do we do that? Well, let's say that this is a strand of DNA right over here, and I'm just drawing it as a line, but this is a double-stranded, and I'll just write it down. This is double-stranded. I don't want to have to take the trouble of keep drawing the multiple strands. Actually, let me just try to draw the 2 strands, just so we remind ourselves. So there we go. This is the double-stranded DNA, and let's say that this part of this DNA has a gene that we want to clone. We want to make copies of this right over here. So gene to clone. Gene to clone. Well, the first thing we want to do is we want to cut this gene out somehow. And the way we do that is using restriction enzymes. And there's a bunch of different restriction enzymes, and I personally find it fascinating that we as a civilization have gotten to the point that we can find and identify these enzymes, and we know at what points of DNA that they can cut. They recognize specific sequences, and then we can figure out, well, which restriction enzyme should we use to cut out different pieces of DNA, but we have gotten to that point as a civilization. So we use restriction enzymes. We might use one restriction enzyme, let me use a different color here, that latches on right over here and identifies the genetic sequence right over here and cuts right in the right place. So that might be a restriction enzyme right over there. And then you might use another restriction enzyme that identifies with the sequence at the other side that we want to cut. So let me label these. Those things right over there, those are restriction enzymes. Restriction enzymes. And so now you would have, after you apply the restriction enzymes, you will have just that gene. You might have a little bit left over on either side, but essentially you have cut out the gene.